Hello and welcome to the BloodStream podcast, a show serving the greater bleeding disorders community, brought to you by Believe Limited and BloodStream Media, and made possible by our presenting sponsor Takeda and our special episode sponsor CSL Behring. I am your patient advocate and host, Patrick James Lynch.
And I am your healthcare advocate, nonprofit nerd, and your other host, Amy Board, reminding you to please speak with a treatment professional? Healthcare
Ideally,
professional.
either one.
Both are professionals.
Just
before any treatment decisions.
Yeah, media professionals is not where your consultation should end.
That's not the thing.
Not at all. On today's show, we have got BDC for you. Amy and Hannah were there roaming around like reporters, living their best correspondent life.
Literally a correspondent.
Just walking around the exhibition hall with a microphone saying, hey, come over here and talk to us, please. We got a camera, and it worked. And you're going to get to see and hear the results of all that on this episode. We've got all of that and more. Welcome to BloodStream.
Listeners, as always, thank you for joining us today. And if you like what you hear, which you will, because I am a roving reporter today, which is kind of fun, you can subscribe to the BloodStream podcast wherever you listen to those podcasts, and you can find us on social media and watch us on social media as well.
I'm afraid we're going to lose Amy to CNN or something if we keep this up. We got to
like true crime, like Dateline.
Yeah, see how excited she's getting? This is making me nervous. Let's just keep doing the podcast. Today, our episode is brought to you by CSL Behring and their hemophilia B gene therapy treatment. To learn more about gene therapy, to hear patient stories, and to view additional support resources, visit beyondhemb.com or download the B Support app wherever you get your apps for more information and to manage your treatment journey.
Thank you, CSL.
Thank you, CSL. What was the best part about being a reporter at the BDC?
It was the cool little microphone things.
Oh, okay.
Josh, who is like heads up our production and things, got us these really cool microphone little things. And so I was able to pull people aside and literally like do the microphone thing.
Did anyone give you the brush off?
No one gave me the brush off.
Wow, look at that.
It was kind of a big deal. And we got some gets, if I do say so myself. We saw Dr. Quon, who I asked how you were holding up, and she's like, HIPAA. It was so fun.
You should know that, nonprofit nerd.
It's great.
Thank you, Dr. Quon.
You bet. I mean, it made it into the episode too. I was like, surely this will be cut. But I was like, so is Patrick like keeping up with what he's supposed to do? You know, just trying to be funny. And she's like, HIPAA.
Nice. See, that's why I like her. She isn't going to let that fly.
No, it's great. But we spoke to folks about gene therapy, specifically for hemophilia B, and we got great folks. We got several patients with hemophilia B. And then to round out this episode today, Hannah actually had a conversation with a patient who's marvelous. His name is Greg. He's part of our older generation, and he's a science nerd. And he just had a wonderful, wonderful, wonderful story. So it's kind of a great episode.
That's at the end?
It's at the end.
Okay, really nice. Well, let's not delay any further then. Here it is. Thank you, CSL, for making this episode possible.
Yes.
And let's get to the segment.
Hey, folks. Welcome back to another episode of Gene Therapy, A Whole New World. It is your girl, Hannah Berry, and man, do we have a fun day planned for you guys. I have my very good friend, colleague, some may even say boss, with me, Amy Board.
Yes, I am. For the sake of this podcast, I will be your colleague.
Oh. Noted. Thanks for joining us, Amy Board.
Heck yeah, excited, Hannah Berry. And this is going to be a great episode for y'all. So today, actually, we're reflecting back on BDC, the Bleeding Disorders Conference, which was in Denver, Colorado in August. And I was able to do some interviews, like a little bit, like a reporter on the scene, if you will.
Yeah.
It was very fun to like grab people and to talk a little bit about gene therapy for hemophilia B in particular.
And we know how busy those exhibit halls can be. So it's kind of a madhouse pulling people off the streets. But man, it was fun. And we heard some really cool things out of it.
We really did. I had conversations with Dr. Sidonio, which was fantastic. What a phenomenal get. And it was really fun. And he's taller in person than I realized.
Who knew?
I really had to look up. Dr. Radak, who actually was on the podcast just a couple weeks ago, which is fantastic, was able to talk to him. and he is marvelous, a marvelous researcher, and he has hemophilia himself.
It's crazy. He's so wicked smart. He could tell you anything about everything, it seems like.
Yes. And then we were able to catch Dr. Quon, who's, it's funny. This is like her claim to fame, but she's Patrick's hematologist.
Right. Not pediatric.
Exactly, an adult hematologist, and anyway, she's marvelous and has other things to her name other than being Patrick's hematologist. But anyway, it was so fun to talk to Dr. Quon. And then we had a couple community members, Matt, who has hemophilia B and was not qualified actually for the gene trial therapy, which is fascinating to talk to him. We talked to Collin, who has mild hemophilia B. Fantastic to talk to him. And then tell me about our last interview, because it wasn't at BDC. It was actually, we were able to catch him on the back end. But tell us a little bit about Greg.
Greg is a 72-year-old man living with hemophilia B. But my goodness, he's an engineer by trade and believes in science. He is a human who will tell you, don't walk, but run to the science. And that just it resonates with me because we need those people to go through the trials so we can keep growing and evolving our treatment plans. So stay tuned. Greg was so fun to talk to.
Oh, it's going to be great. So that's going to wrap up our episode today. But let's get into it. We had fun at BDC. Let's get into it. I think our first interview that you're going to hear is with Matt, and he shared a little bit about his anxiety about getting stuck every week for Factor and how that played into his decision to maybe test the waters to see if he would be eligible for the gene therapy trial.
Right. Let's kick it off with Matt.
BloodStream listeners, we're at BDC, and guess who I found?
Hello, how are you? I'm good. I'm glad to be here.
Now, this is your first BDC, correct?
Yes.
Tell me everything.
It is a great flight to get out here. It's great to see everybody. I'm looking forward to some of the sessions and, of course, the movie.
Of course. Dismissed.
Yep.
Going to Dismissed premiere tonight. You can't miss it.
Yep.
Matt, tell me, first of all, what type of hemophilia do you have?
B severe.
B severe.
Yes.
Tell me a little bit. So we're asking some people when we find folks here a little bit about their bleeding histories and breakthrough bleeding especially. Tell me a little bit about your bleeding patterns throughout your life and what breakthrough bleeds have been like for you.
Growing up, my breakthrough bleeds were the ankle and then the left elbow. I've lost motion in the left elbow. I can't flex it all the way. And eventually with my ankle and the breakthrough bleeds, I ended up getting my ankle completely fused. So it doesn't bleed anymore.
That's
I can't move it.
But you can't move it.
Yeah, exactly.
Now, would you describe, as someone who doesn't know what breakthrough bleeds are, would you consider a bleed and breakthrough bleeds like those in-between bleeds? Or are you bleeding while you're on prophy or treatment? How do you describe your bleeding?
I've been on prophy for many, many years.
Yeah.
So the bleeds in the elbow are gone.
Okay.
I get the occasional bruising here and there. I'll wake up and the girls will be like, "You have a bruise on your back the size of a softball." Like, I don't know how I get it. It just happens. But with the prophy, it helps with the nosebleeds. It helps with, if I'm up walking around and working, it helps with the left ankle because that one's starting to get bad, and the prophy helps calm that down so it doesn't just pop out and ruin my day.
Right. What has the treatment journey been like for you? I mean, there's so many more options now. And what does it kind of look like, all of these different arenas?
Well, I've been on prophy in multiple companies, Benefix, of Holix, Alpha 9, Retubis, that's run Revenine right now, and I'm also on Hempazit.
Okay.
So I use the Revenine for any breakthrough bleeds that happens in between my injections.
Okay.
And I actually had to do one last night.
Oh.
I had nosebleed. I'm like, I'm not messing with this. I'm going to take care of it right now, and it stopped. Wow. So it works out well. I have both medicines on hand, so I'm taking the Hempazit prophy, and then I have the Revenine for backup.
Now, I know you were interested in doing a gene therapy trial.
Yes.
Before we kind of talk about the outcome of that trial, why were you intrigued about gene therapy for hemophilia B?
the anxiety of getting stuck every week. It was just getting kind of, I don't know if I want to do this anymore. And the veins were getting pretty bad.
Yeah.
So I just decided this might be a good option to help me long term.
Right.
And I don't have to infuse once a week or twice a week. And help just with my busy schedule, keeping everything organized. I just take the one medicine and knock on wood, I would have been good for years and years without having to take Profi anymore.
So that was Matt. Excellent point of view, I think, and really an important point of view, especially with this community. Hemophilia B is much rarer than hemophilia A, which we all know. So it's a smaller collection of patients, and so stories really matter. Those archetypes really matter. Right. So it was really good to hear from Matt. But let's transition into our next guest.
Oh my gosh, Dr. Sidonio. I can't believe we caught him still.
I was about to say, kind of a get.
Huge get. Let's go.
Right. So.
We're live on the scene. You guys were live on the scene. We caught Dr. Sidonio, who's one of my favorites. Hi.
How you doing?
I'm doing well. How are you? It's so fun. We finally get to meet in person.
Yeah, I'm doing well. I got a lot of sugar from the exhibit hall, so I'm feeling pretty good. So,
what was your favorite exhibit hall treat?
Santa Fe's ice cream cookies are pretty good.
Sure.
Did you get to rotate them in whatever topping that you want?
Yes.
So yeah.
Does anyone have a coffee cart this year?
That was the issue. That's my only complaint.
It's like no coffee cart. Usually there's like 16 coffee carts.
That's gonna be my only complaint on the comment card is you need more coffee. So yes.
Did I feel like you're closer?
Okay. Okay.
All
right.
I feel awkward. Now I feel awkward.
That's okay.
No, it's totally fine. Well, let's get into it. I just I have science questions. Okay. Thanks. So one of the things that we're most interested in right now is breakthrough bleeds, at least for me. Just kind of like nerdy. Can you explain what a breakthrough bleed is?
Sure. Yeah. So in patients, we're talking about in the context of a patient with a bleeding disorder.
Right.
And so ideally patients that would have no bleeding at all, but we know that at times when they're in their natural state, it could happen either spontaneously or induced by trauma, or it could be in either one of those settings. It's where they have an acute bleeding event, and it obviously disrupts their lives in some way, and it may or may not require intervention, because there's some bleeding events that can be observed. Maybe they can do non-bleeding measures, like a nosebleed might go away if you put one of those fancy nose buds on, if you stop your activity and cool off. Or it may require intervention with an antifibrinolytic or even factor therapy. And so these are events that happen, all the time in a patient's lives that have a congenital bleeding disorder. So they're just bleeds. They just call them bleeds.
They just call them bleeds.
Yeah.
They're just bleeds.
They're just bleeds, yes.
With these new novel therapies that are coming out, especially we're thinking about gene therapy, hemogenics for hemophilia B.
Sure.
What has it looked like in terms of breakthrough bleeding on hemogenics?
Yeah. So, there's been some concern that there's been a general lack of interest in gene therapy by the patient community, by the physicians that were all demanding that we get this. But, this study has really delivered and has demonstrated that patients, can go through this trial, receive a single injection. And if you look at the average levels, these patients are in the sort of near normal range. They're not normal, but they're in the sort of high, mild hemophilia range. And because of that, these patients are off prophylaxis with factor therapy. They were all on either standard half-life or extended half-life. I think most of them were on extended half-life therapy. But they only have to get on-demand treatment, which is rarely happening.
Wow.
So which is really good. And so they really did. So initially the company was Unicure, and then CSL purchased it, but they did a really good job of conducting the trial, which was during the pandemic, which was not easy. And then, the data has shown that 18 months later, 24 months later, they're still doing really, really well.
That's incredible to hear. And have you heard from patients that they understand this data, that they know about this data?
Yeah, I think they definitely know about this data. Probably don't know about it as much, because maybe it doesn't get talked about in this context of these kind of meetings. And there have been some updates where people do update that. And so I think people know that it happens, but I don't think they quite understand the impact of this that it looks like it could potentially be a very long lasting, elevation of the levels. We don't know if it's going to forever stay this way, but like during one of the presentations, when they look back at some of the data from 15 years out from original studies, there are patients that still have expression. It's not as high as in these new studies, but they're doing really well. And so we expect these patients to have elevated levels for many, many years. And so I think that's really exciting. The adverse event rate has been very, very low. We obviously document every little thing that happens. Somebody gets a runny nose, it gets documented. Somebody, has a bad night drinking, it gets documented. They shouldn't be drinking, so definitely not drinking excessively. But any of those things get documented, and we haven't seen any change in the safety signal.
That's incredible. Tell me a little bit. This also is, I feel nerdy about this, and I don't know how much data is out there.
Yeah.
How is gene therapy shifting the treatment landscape when we're preventing long-term damage, particularly joint damage? I realize it's so early, but what are you seeing?
Yeah, I think, obviously the first step to preventing long-term damage is to stop the acute bleeds. All these breakthrough bleeds have to stop.
Right.
And so clearly when they've elevated the level, it's not enough just to elevate the level in hemophilia. We need to elevate the level and completely eliminate or reduce down to near zero the bleeding events. So once you've done that, then many of those long-term effects could start to at least correct. You can at least bend the curve. Because if you're older, you're 30, 40 years old, you certainly will have damage that you've accumulated in your first 30 years of your life.
Right.
Of breakthrough bleeds. And there are certainly patients that had target joints or multiple joint bleeds coming in. But certainly the hope is that they'll continue to march down in age, offer this to younger patients that have healthier joints, and we may be able to completely reduce the amounts to very small amounts. I don't think we can completely eliminate this. And the reason I feel this way is because they've raised the level into a very mild hemophilia range. And we know based on our data on hemophilia carriers that they still have some minor joint disease, not at the level of somebody with a severe hemophilia, but they have a level of joint disease, certainly when you compare it to normal women.
Right.
You have these big study from Sweden that demonstrated this. But certainly the amount of bleeding is prevented is significant. So we're hoping that we're preventing any GI bleedings that will happen in the future. Hopefully the amount of joint bleeds will be very delayed. When we first started doing prophylaxis a long time ago, not when I was doing it, but when some of these older docs were doing it, we were just trying to delay it till their adulthood.
Right.
Now we're trying to delay it until their elderly age and maybe even in the future delay it completely where it doesn't even happen.
Right.
So that's what the promise of gene therapy has, because you just don't have those ups and downs that we do with factor therapy. So.
Right. It's so fascinating. Thanks for taking the time to do this. Let me look over my list and see because you're so eloquent. One question I want to ask you, what should patients realistically expect in gene therapy?
Yeah, I mean, I think, the one thing you don't want to do is for patients to come in with unrealistic expectations, thinking they're just going to walk in the door, stick their arm out, get the IV, walk out, and then say, see you in 3 months.
Right.
So there's obviously a commitment. A couple of those things are, delayed family planning. There's a commitment to getting labs, on time, making sure you're available to get those labs because it's a responsibility. This is a fairly expensive product. And it's important that, they take this responsibility because all of it could go away.
Right.
If there's a potential concern for a loss of expression. And so I think they just have to understand there's a commitment early on. And over time, the amount of labs that you need really, really gets minimized where you're over time, after about 6 months, you're really seeing the patient as much as you normally would. So you have to have good communication. And this is where I think, when we have patients that undergo bone marrow transplants and things, they're not like binding contracts, but they're these patient contracts that are written out where the expectations are there. It's like when you need to respond to your text. You need to respond to emails. If we call you and say you need to get the labs tomorrow, you really have to try everything you can to do this.
Yeah.
And then you really need to commit to coming back to the clinic. Because remember, not all these patients are patients at our center.
I know.
They could be traveling 4 or 5 hours.
No.
And so, it's a burden. And so also the commitment is with whatever center you're working with, that you're going to work together to make sure the patient is safe. In that early part, there's a potential for loss of expression. There's a potential you might have to go on a short course of steroids. But, so we need to make sure. So when we say that, the AST or ALT are elevated, we need to repeat this. It can't be like, we'll get back to you next week. It's got to be a fairly good response. And so I think the commitments are important. And so you have to know in your life if that's the right time for you, right?
Right.
And so it might not be the right time right now, but 6 months from now, it might be the right time. If you're planning on having children in the next 6 months, this is not the right thing for you right now,
Right.
but certainly can do it in the future. So
Cool. Were you excited about it? I mean,
Yeah.
it's cool.
Well, no, it's very exciting. And we had a session earlier talking about the next wave of gene therapy products that are available. And so it's great that they're not just starting with this, AAV-directed, liver-directed gene therapy, that they're really thinking about other options. And they're looking at things that are happening that we can't sort of fix. There are more issues on the factor VIII side. And they're saying, okay, maybe if we do it this way, we can eliminate that problem. And so, obviously the science is going to be really important. It's the only way it's going to sort of advance this. And obviously the goal is to be normal. Right. So right now we're accepting, getting to the mild hemophilia range. It's great. These patients are doing well. They're off prophylaxis. That's a really good thing. And so, and they're living their lives and, doing those things. And it's funny, you hear these stories. I remember one of the patients told me, I had that excuse where I told my wife I couldn't do all that kind of yard work because I have severe hemophilia. Now I don't have any freaking excuse to do it. I have to go do these things. I can't say, I can't do it. I say, well, your levels are pretty good. And aren't you doing really well? Aren't you sort of in your levels near normal? And so, so I think that's important. And it is good for the patients because we want them to participate in life. We want them to exercise. We want them to do those things that they were limited, from doing before. So,
Well, thank you so much for joining us on the BloodStream, Dr. Sidonio. He's kind of a legend, but thank you. And it's good to meet you in person.
Yes, more than just on.
I know.
Always love talking to Dr. Sidonio, in particular about gene therapy. We haven't had a conversation with him about gene therapy for hemophilia B in a while, so it was really lovely to hear his thoughts.
He's been on a lot, too, doing a lot with gene therapy. So it's cool to hear kind of the beginning, the middle, and where things are going with it.
I agree. He's fun. So the next community member that we were able to speak to has mild hemophilia. And what a fantastic perspective when thinking about gene therapy, because he had terrific perspective about it.
Totally. And I mean, we know when treatment works, it works.
Right.
And there is that mindset of why fix something that isn't broken.
100%.
And when it works, it works. But we know mild, moderates, there's still pain. There's still joint pain,
Yes.
symptomatic bleeding.
Yes.
Not symptomatic bleeding.
Yes.
Is that the word I want to use?
Let's go back.
Spontaneous bleeding.
Yes.
That was the word.
Spontaneous bleeding. No, totally. And, and I do think a lot of folks with mild and moderate hemophilia have told me that they downplay their symptoms a lot. I'm not severe, so I shouldn't worry about it. But when you have severe hemophilia, 9 times out of 10, you can really pinpoint that this is a bleed or this is this pain, this arthritic pain, is really from a bleed or from my hemophilia. And with mild and moderate hemophilia, it's harder to navigate.
Totally. And so then are they still in that wait to see to treat?
Yes.
We know that language is shifting. So
Yes.
let's see what Collin has to say about it.
Everybody, listeners, I'm here with Collin, hemophilia B patient. Is that right? Here at BDC. How many BDCs is this for you? 9,000 or you're second?
I'd say probably 6 or 7. It's been a while. It's been 2 or 3 years since I've been to one, but
Now, your mom
good few.
is very well known in the community. Did she get you into this? Is this the whole thing?
We kind of got into it together. We started getting involved when I was 15 or 16 and just started doing a bunch of stuff together. And I've kind of just both forged our own ways together and on our own a little bit. So.
Collin, tell me a little bit about life with your hemophilia. What has your bleeding been like? What were your breakthrough bleeds like? What has it been like?
So I'm mild and blessed to not have many problems, thankfully. I definitely have had some joint issues over the years and have a little damage, like I think all of us do. But overall, I'm able to just live a normal life. I do a lot of hiking and climbing and slacklining and all sorts of crazy things, and snowboarding. And so I try to stay super active and do as much as I can. But I think that with treatment these days, especially, it's just even easier now than it's ever really been.
What has it been like? Because now with all of these new novel therapies that are out, there's a shift where the severes are starting to have levels like they're mild or moderate. So the bleeds tend to be more spontaneous, which is something that you've lived with your entire life. What has that been like, kind of having more of a community understand what the bleeding pattern is like as a mild?
It's been interesting because I see a lot of people talking about, oh, well, I can get gene therapy and I'll be mild and never have to infuse again. Obviously,
Is that true?
We know that
Okay, it's okay. Is that true?
But I think that, yes, it's great for a lot of people, of course, and, cutting down on infusions is awesome. But I think that it's really important that we don't promote, hey, you're not going to have to infuse again.
Right.
You still need to have this skill and you're willing to do it and ready to when they need to. But it's great that it's less, yeah.
Well, I also think there's a perception that if you're mild, you have fairly healthy joints. And is that true? Do you have joint damage? Do you have pain?
I'm a little on the fence. I have pain, yes.
Okay.
But I've had CAT scans and ultrasounds and all. It doesn't look like there's any damage.
Yeah.
So I honestly don't really know exactly where it's coming from.
Right.
But I definitely do have joint pain. So.
Now, I know gene therapy necessarily isn't an option for someone with mild hemophilia. Is that correct?
I believe there is one out now that you can be approved for being a mild, but it seems to be under fairly specific situations. You're having a lot of breakthrough bleeds and things like that.
What does something like gene therapy, a treatment like gene therapy, look like for you in your lifestyle? Is that something that's interesting?
Yes and no. I think that so my levels are around 13%. So I think realistically that could boost me up to probably having almost no issues. But I also am at the point where I tend to only infuse maybe once a month, once every other month. So at that point, the risk of gene therapy just hasn't been out that long.
Right.
And we just don't have all the long-term studies and things. So I think for me personally, with how little I'm having to infuse in the first place, that the risk outweighs the reward a little bit.
Awesome. What, looking to the future, what are you most excited about in the treatment journey? What would be something that would be really special for you in your management of hemophilia?
That's a good question. I don't know. I'm pretty satisfied with my treatment. I am on a standard half-life product, and things that just work. It works great for me. So I don't really have any there's this one big thing that if that was fixed would solve everything. It would be nice to not infuse a pill or something would be cool, but I just don't see it working the same or being as good. Or I'm sure science can get there eventually, but we'll see.
You heard it here first, everybody. We got to get that pill. Thank you, Collin. Thank you so much for joining us, and have a good BDC.
Yeah, thank you.
So thank you to Collin. Again, that was awesome that he stopped and chatted with us. He's from Colorado, which both of us are from Colorado too. So
hits the heart.
Hits
the heart.
Rocky Mountain High. We love that. We have Dr. Quon next.
And Amy trying to get a little silly here. And you'll see. But the most important thing is HIPAA's real.
There was a lot of takeaways, some great bullets from Dr. Quon, but also HIPAA is real.
HIPAA's real.
Let's take it away.
Hello, Dr. Quon. It's so lovely to meet you in person.
Hello.
I
Nice
see
to be here.
Your claim to fame is that you were Patrick's pediatric hematologist.
Not pediatric. I'm his adult hematologist.
So you can give us all the down low on Patrick's stuff? This is great.
That might violate HIPAA.
I might violate HIPAA, But I would love to ask you some questions about Patrick's joints. I'm kidding. No. I do want to ask one thing that we're a little nerdy about lately is this concept of breakthrough bleeding. And with all these new novel therapies, what the heck is happening with like breakthrough bleeding? I guess to like simplify it, what is a breakthrough bleed? Is it just a bleed?
So breakthrough bleed can be any type of bleed. So as you might have heard in hemophilia, the majority of the bleeds are into the joints.
Right.
So if you look at some of the statistics, they say, 80 to 90% of the bleeds are into the joints.
Right.
And so when we look at joints, we focus, the health. And when they talk about bleeds, they include all bleeding. So that it can include muscle bleeds, that can include soft tissue bleeding, maybe retroperitoneal bleeds, so internal bleeding. All of those are considered bleeds.
Right.
But joints, there's breakthrough joint bleeds. They break it down into, when you look at clinical trials or different clinical trials, they break it out into all bleeds and then joint bleeds. So I don't know if that answers your question about breakthrough bleeds.
No, it does, because I think like the bleeds or the bleed count, the bleed number, how many bleeds a patient has a year, annual bleed rate, those things are so important from a data perspective. Is that correct?
Yes.
Okay.
And so they do break it down into all bleeds,
Right.
then treated bleeds, then joint bleeds.
Okay. Oh, it's like a whole thing. There's like all these different categories. I didn't realize.
That's the bleeds.
Okay.
Yes.
Okay. So now, we used to, it felt like it was a little bit more black and white back in the olden days, the prophy, replacement factor therapy days.
Yes.
You have a bleed, you treat. You have a bleed, you treat.
Yes.
Now with these new novel therapies, what has it been like to treat bleeding? Yeah.
It depends on the therapy.
Right.
And so some of the newer agents, which we were discussing earlier today, your treatment of bleeds is still with factor therapy.
Okay.
So factor concentrates.
Okay.
If you don't have an inhibitor and you're hemophilia A, you use factor 8 concentrate. If you have hemophilia B, you use factor 9 concentrates. And then we can spend another 20 minutes talking about all the different factor concentrates we have to use.
Right.
But a factor concentrate to treat the bleed.
Right.
And that's not including inhibitors.
Right.
If we're talking about those.
Right.
So those are different concentrates. But again, it's factor concentrate to treat a bleed that happens if you're on a rebalancing agent, if you're on emicizumab, etc.
I know we don't have a lot of data. What is the data showing with the hemophilia B for gene therapy, with hemogenics, and breakthrough bleeding, joint bleeds? What has it been like for those patients that have been on gene therapy?
So they did a study when they did the gene therapy. They did a study before they went on to the gene therapy. So there was a before part where the patients kind of logged all their bleeds. And then after they got the gene therapy, they continued to log their bleeds. And that number actually went down. I don't have the exact numbers, but their annualized bleed rate wasn't 0, but it did decrease by over 50% is my recollection.
Okay. Well, that's pretty. I mean, that's
good.
That's good.
That is good.
That's really, really good.
So the gene therapy seems that the patients are making factor on their own after the gene therapy and preventing the bleeds from happening.
Science, you guys, science. No, that's great. That's really great. What do you think about the future of durability with some of these novel therapies, gene therapy in particular? What are something that you're hoping for as a physician to, for gene therapy in particular?
For gene therapy, it's a little different between hemophilia A and hemophilia B. So the durability in hemophilia B and that factor 9 gene therapy seems to be durable. So the levels they got in their first year is similar to the levels they got in their second year, and then now out to like 4 or 5 years, those levels are pretty stable.
Okay.
It's a little different than in hemophilia A, where the first approved product.
Right.
The levels were higher in their first year,
Right.
and then it took sort of a drop in the second year. Then it started to stabilize out in the third, fourth, and say fifth year.
Right.
So the steep slope, if you call it, of that drop is stabilizing out.
Okay.
So between the first year and the 5 years that the gene therapy has been in the trial and being looked at, it seems that the hemophilia A is not quite as stable in between the comparison of year 1 and year 5, but between years 3 and 5, it's more or less stable-ish.
Last question. What should I bug Patrick with? I'm kidding. I'm not gonna I was just gonna be like, so what should I make Patrick do with his treatment? I don't know. I got nothing. That was a terrible
That
joke.
is a shared decision between Patrick and his doctors.
Signing off with Dr. Quon.
Yeah, and it may not be with me, but it should be with him and his doctor.
That's a true professional right there. Thank you so much for joining us and all the things. Listeners, Dr. Quon, thank you so much.
Dr. Quon, we love. What a joy. She's wonderful.
And funny.
And
love
funny.
some humor.
Yes, and just wildly smart and can speak the patient speak so well.
Yes.
Yeah.
It's fun to sit down and chat with her.
Yes.
Makes a difference.
Over and over and over.
Totally.
Which actually makes a wonderful transition into our next person that we were able to grab.
Dr. Radak.
I know. Good old Dr. Radak Kazmarek, who does not see patients. He's a researcher, but he focuses on gene therapy.
Yeah.
And he has hemophilia himself. And so he just has like a fantastic mind for where the science is and where the science wants to go. So if you're really nerdy into this stuff, you're going to love this.
Yeah, this is our nerd piece.
Yeah. Yeah. This one's for the nerds. Take it away, Dr. Radak.
Okay, BDC listeners, all the things. We're so excited. We're here with Dr. Radak. Thank you for joining us.
Thank you.
Thank you for absolutely love that I got to pull you aside. I wanted to talk to you a little bit because you are a bit of a rare duck in our community. You are a patient with hemophilia, is that correct?
That's correct, yeah.
And you are also a researcher in hemophilia and bleeding disorders. What is your primary focus in your research?
My main focus is immune responses to gene and protein replacement therapies for hemophilia A and B. I've been involved in multiple facets of research from basic bench science to translational science to clinical research, having been involved in several clinical gene therapy programs for hemophilia.
Were you involved in the hemogenics studies at all and kind of hemophilia B in that land?
I've been involved in the analysis of the pre-existing anti-AAV5 study as far as this one. So, yeah, on that front.
What have been kind of like the standouts of that study to you that have been really interesting, specifically for the hemophilia B community?
One interesting feature is that once you get tested negative before actually getting dosed, so when you're considering getting the therapy and your eligibility has to get checked first, we need to check if there is a pre-existing immunity against the vector, which means if it's there, the vector most likely will not work for you because the pre-existing anti-AAV5 antibodies, AAV5 is the serotype that the gene therapy uses to deliver the transgene encoding the therapeutic protein to your liver cells. If you have those antibodies on board, they will prevent the vector from getting to those target cells, and so we won't be able to benefit from the therapeutic effect. So if you test negative, it means you don't have those antibodies, and that's one of the things that's off the list to check and clears you to actually get the therapy. Yeah.
Interesting.
And if you one of the key takeaways from the study is that if you test negative, and there's always some space between actually testing and actually getting dosed with the vector, so you are unlikely to get infected with a wild type AAV5 virus, which would give rise to those pre-existing antibodies that would then prevent the therapy from working. That's very important because, I mean, there is this, because potentially those antibodies, if at screening you test negative, then you get infected, you develop those antibodies, they may rise, their level may rise to, they may rise to a level that will be problematic and could potentially impact the therapeutic effect negatively of the therapy and perhaps even prevent it. I'm qualifying this because for hemogenics, one thing for which this therapy stands out across the board from all the other gene therapies, the approved ones and a number of others in the clinic, is that pre-existing anti-AAV5 actually is not an exclusion criterion for you to either enter the clinical trial back in the day or now to avail of the product, because from the phase 3 clinical study we actually know that you can have a pretty relatively high titer of that anti-AAV5 antibody and still benefit from the therapy. This is unique. In all other clinical studies we have not seen that, maybe with the exception of one hemophilia A study where some individuals actually did, we call it seroconverted, so develop antibodies between screening and dosing, and they still actually responded to the therapy pretty well. So there's something special about the serotype used in the case of hemogenics, AAV5, that makes those anti-AAV5 antibodies more permissive of transduction, of entering the cells and making them make the coagulation factor.
Interesting.
Yeah.
I mentioned it. Me? Okay. Thank you. Is that good?
Am I speaking loud enough? Yeah? Okay.
One more step. Sorry, they're trying not to get logos in the background. God bless. Thank you. I mentioned it in the beginning. You are living with hemophilia, and this is your research focus. What excites you about the potential of gene therapy in the future for hemophilia in particular? What gets your nerd, nerves going?
I would say that, well, the fact that the gene therapy is the only intervention that comes so close to addressing the root cause of the disease. And that means it's uniquely well positioned to provide a cure, basically a functional cure, which would mean, in my opinion, opinions vary there, but in my opinion, a functional cure would mean somebody achieving a long-term, ideally lifelong factor 8 or factor 9 level that would make them not need any additional hemostatic therapy whatsoever, and no matter the circumstances, including a surgery. Yes. So that's a high bar. And actually, some individuals with the current gene therapy, some of them achieve that actually. Some individuals do land in the normal range or upper mild range, who would really basically, would offer that hemophilia-free mind that we are after. But then not all individuals, because there's wide variability. It's quite wide in the case of hemogenics, and it's much wider in the case of hemophilia A gene therapies. Having said that, still 1/3, if memory serves, of individuals in phase 3 study of hemogenics, 3 years after dosing, they had normal factor 9 levels. So that's a tremendous achievement. So that really is something amazing for me to think about from the perspective of living through several different generations of treatments, and remembering the times when gene therapy was becoming a thing in terms of a concept, that once we learn how to make recombinant factors, that gave rise to the concept of gene therapy, because recombinant factors, the way we make them, we put a gene encoding factor 8 or factor 9 in the cells in artificial conditions in a big bioreactor in a tank.
Yeah.
Right? We control everything there. But once we became able to do that, the idea arose, if we can do that, maybe one day we'll be able to put that gene, instead of putting it
putting it in those cells in an artificial cell culture, maybe we'll be able to actually put the gene somehow into the patient's cells directly in the patient's body and make the body make
the body do it.
protein, right?
It's incredible.
So the journey, the decades of experimentation, fits and starts that we've gone through, and that we are actually so close to it, even though there's still ways to go. It's a work in progress, but we're getting there. This is something that really gets me going.
Yes, it's like the best soundbite of the day. It's so great. One more question I would like to ask. Tell me a little bit. Tell me a little bit about breakthrough bleeds and joint bleeding, specifically on hemogenics. We're really, kind of focusing on the hemophilia B gene therapy. What does the data show? That's kind of a lot of murmurs in the community, specifically about breakthrough bleeding on novel therapies. How do we treat? Do we know? I'm scared to do it because we don't know how to treat. What are your thoughts? What is the data showing about breakthrough bleeds and joint bleeding in particular with gene therapy?
Well, the data from clinical trials show that hemogenics is, not inferior, and that is actually superior from standard treatments, which means it further reduces bleeding rates. And that's compared to standard treatments, meaning prophylaxis with standard treatments, right? So, which already has, already reduces bleeding rates to really low single-digit numbers, right? In the case of gene therapy such as hemogenics, we are getting to median of 0 bleeds, right? So in terms of breakthrough bleeds, again, because of the variability, I think they will be, it is a problem for some individuals more than others. Those that land in upper mild or normal levels of factor IX, they're unlikely to have breakthrough bleeds. And I guess in those cases, we are more likely to potentially mistake something else for a bleed. Although I'm very cautious about saying that, because then it happens too often that things that potentially could be bleeds, subclinical bleeds, get dismissed because, maybe it's arthritic pain or something.
Right.
It could happen.
Right.
But I would be very, very cautious about dismissing it too soon. But still, at a higher factor level, normal factor level, you are very unlikely to actually experience breakthrough bleeds. Those who land at lower levels potentially might bleed. And 2 individuals, if memory serves, in phase 3, one did not respond to the therapy, one with a very high level of the anti-AV5 antibody. Again, it's one individual, so we're not even 100% sure that was the cause. That's the most likely cause, because his titer was really high compared to all the others. That was the only one with pre-existing anti-AAV5 who did not respond to the therapy. Everybody else did respond very well. So I want to say that. And then one individual did not respond at all because he only received 1/10 of the dose that he was meant to receive, because there was some infusion reaction. So those individuals returned to prophylaxis. But in terms of people who do achieve some level of therapeutic response and potentially may experience bleeding, I guess it's an easier decision to treat them as opposed to other novel therapies that we've seen being rolled out. And because here we have factor IX on board.
Right.
And we are basically, if we need to infuse with factor IX concentrate, we're basically just topping up.
100%.
Yeah.
Okay. 100%. And with other novel therapies, some of these rebalancing agents, it's much more difficult to know, what do you do? Okay. Oh, yeah. Okay. You're right. That feels very black and white. Well, thank you. What are you doing tonight? Do you guys?
I do have dinner plans.
All right.
And we'll see. It sounds exciting.
That sounds fantastic. Thank you for joining us, and I hope you have a good rest of the conference. And I look forward to having you back on the show to, have you talk a little bit about what you're discovering and more of your work. So thanks.
Always happy to come on. Thank you for having me.
Thank you.
I appreciate it.
Thanks.
Do we treat? Do we wait?
Yes.
Do we see?
I know.
We've always had these questions, but it feels like it's peaked again.
I 100% agree. And something that when we were going through the footage here, we saved this piece of Matt's interview specifically for this section because I think his determination to be involved in this science for the next generation, which includes his daughter who has hemophilia B, is really great. Shout out to Izzy. We love her. But I think his liver health really played a piece in his...
Yeah. We might have the desire for it, but, there's always these outside factors that come into play. And so it's important to know and to still try.
Absolutely. So here's this piece from Matt. Tell me about the outcome of being on that trial or trying to attempt to get on that.
I tried to do it. I jumped through all the hoops.
Yeah.
I found a lot of good things, bad things about my liver. With the gene therapy, you have to have a perfect liver. And I had a lifestyle earlier before Isabel that wasn't so healthy for my liver. And I actually went through the liver biopsy, and they were like, no, it's not going to happen. Sorry.
What are like thinking, not for yourself, but for the future generation of hemophilia, having gene therapy as an option, as it's just, the science is getting better, it's getting more durable. What are your thoughts about gene therapy for the future?
I think it's a great step forward. I wish they would have done more tests, more in the clinical trials on women. That's just the way it is for me. I got to look out for my daughter. And my first thought was, I get on this. Of course, it's going to be a trial. They'll study. But hopefully this will be good enough for my daughter down the road. That's why I wanted to do it.
Right.
Is to show her that, hey, this is possible. Because she's taking Pro-Q once a week as well. So I was hoping that this would be a good option for her.
Yeah.
But it's decades down the road.
Well, thank you. Thank you for stopping by.
Yeah, not a problem.
And I'll see you tonight at the premiere.
Yeah, absolutely. Wonderful.
Wow, I tell you what, Amy Board, that just resonates because we were saying, it's for the future.
Yes.
We have to try now with science. We have to be involved so that way we can continue to evolve. Only reason we're now here to gene therapy.
100%.
So cool.
100% so cool.
I tell you what, it has been such a fun episode, but we did promise to send us home with one really impactful message. So let's hear from Greg, a gentleman who has severe hemophilia B all his life. And, well, he has a journey that I'm excited for you guys to listen to. So let's hear it from Greg.
I have been a private person my entire life. I don't like publicity. I don't like attention. I have worked behind the scenes most of my entire life as an engineer to try to improve things. I only reach out now and put myself out here simply because I believe in this, and that the fear would be a horrible reason not to take advantage of science this wonderful. This is not a cure. It's a long-term treatment, but by gosh, in my lifetime, I doubt I'll find anything that comes closer to taking hemophilia off of the list of things I need to worry about, of things that threaten my life. So I would say that especially for a young person with a nice young liver, they absolutely look into this. And in my opinion, you don't walk, you run to science like this. A lot of us have gone before you in order to make sure it's safe. It is safe. I think that all the indications are that it is safe. I was born before there was a diagnosis for factor IX. Dr. Quick from the University of Chicago was involved at my birth, and he did diagnose me within a few days of my birth that I was a factor IX Christmas disease a person. He had just weeks before diagnosed Stephen Christmas, and who the disease is named after. And so I was one of the very early ones to be diagnosed, and I was diagnosed pretty close to my birth. To put it mildly, a horror show. I was 30 years old by the time I was 2. So it was a kind of thing that grows you up pretty quickly. I was a severe hemophiliac. I am or was a severe hemophiliac. And as a result, I had spontaneous bleeds all the time. My life was defined by the bleeds that I had, when I had them. And so as a result, most of the bleeds that I had younger, I just toughed through them. if they got intense or very severe, they would give me fresh frozen plasma, and that would mitigate the bleed to some degree. But most of it all was, well, here's your Percodan or your Darvons, and go home and try to survive this. And so up till the age of 18, everything was reactive, never proactive. And then cryoprecipitates were used in a reactive manner for probably the first 10 years that they were out. It was in my 20s, late 20s, before they started doing prophylaxis, where I actually took factor before the bleed in order to prevent the bleed. And so a lot of damage was done in that 20 some odd years previous to prophylaxis. Not necessarily a single bleed, but just multiple, hundreds of them, thousands of them. Each bleed took a toll on that joint. And so I bled into my elbows, shoulders, knees, ankles, fingers, everything that you can imagine. Those bleeds were less life-threatening because they were into a joint. The bleeds that you had to be very concerned about were ones like just falling on your ass. You could bleed yourself to death in a cavity like that. And so there were certain bleeds that I knew even as a young person that you just don't mess around with because they could take you out. But the joint bleeds just accumulate over time, and the joint becomes more and more arthritic. Hemgenex was not my first miracle. When it became available, it was just a no-brainer to me that I'm a scientist by trade. I'm an engineer. And so when I saw the science behind it and what they were doing and the fact that it was so well handled by the clinical volunteers, that it was just a race to the line. But what was funny about this, it was CSL that really is a result of me having gotten this done, because I had kind of given up on a shot of this caliber being available to a 72-year-old man. I just couldn't imagine a situation where my insurance company would say, yeah, that's a good option. We'll do that. And so I really was kind of blasé about it. I said, well, this is wonderful for the young hemophiliacs. I just don't see how it's going to help me. And I better be glad that I've got a Delvion and get on with my life, right? Well, they came to me and they said, well, we think that you are a good candidate for this treatment. And why don't you let us worry about the insurance company? Because we have a team of people that know how to work with insurance companies, and let us intervene and come in and see if we can't get this approved for you. At which point you say, well okay, yeah. I'd never had a team of people coming in and advocating for me for anything. And so it was fascinating to watch them work, and to eventually, it took a month or two for them to negotiate back and forth. And pretty soon my insurance company went, okay. And I got accepted and got the shot on September 11th last year, which was a pretty easy date to remember. And it's just been, I had, because I'm old and had an old liver and had hepatitis C most of my life from the factor, which was another clinical trial that cured that and made my liver healthy enough to be able to accept Hemgenex. It all seemed to line up almost miraculously, and I got the shot. It was the infusion of Hemgenex. It's a treatment that I did not really expect to see become available in my lifetime. Science came along a little quicker than I thought it was going to. It was a no-brainer. I kept expecting it to have some kind of effect over me, and there was none. It just as well been a bag of saline that they gave me. It had no physical reaction whatsoever. A little later on, they had to give me some drugs to keep my liver happy, because it's an infection to the liver that delivers these fragments. But that was no big deal either. And my factor levels went up to normal. They went up to the top end of normal. And then as my liver settled down and everything kind of relaxed, my factor levels balanced right there in the middle of normal and have stayed there and haven't budged. It's just a miracle. To me, it's a miracle. To everybody else, it's science, but to me, it was a miracle. And it made such a difference. I never realized what the issue with doing prophylactics was. And that was the hills and the valleys. The hills and the valleys. Okay? Well, when you hit the valley, and everybody hits one, not time to factor up again, things happen. Maybe you don't have a major bleed in a joint, but there's little micro bleeds that you seem to have that caused inflammation, and arthritis started setting in. And my arthritis, you could tell when I could tell you when my factor levels were low long before the blood test could. And it's simply because of the arthritic feeling that I got in all those joints, but at the low end of the spectrum. Well, guess what went away permanently once your factor levels came up and stayed in normal? That evaporated. And suddenly, what I thought was chronic arthritis I was going to live with my entire life, I lost about 50% of it. I still have arthritis. I'm going to have that forever. But this nonsense that I had from the prophylaxis varying up and down just completely went away. And I hit a steady state now where I feel the same all the time. And it's far better than I've ever felt in my entire life.
Well, folks, I tell you what, this might be my favorite episode yet. Not only are we a full-length feature on BloodStream Media.
We are a full-length feature on BloodStream.
Shout out.
Yes.
I have Amy Board next to me, and that is just so fun for me. It's been great. It's been great. So thank you, Amy, for joining us today.
Well, thank you, Hannah. And thank you, CSL Behring, for sponsoring this full-length episode and for letting us go to BDC and finding people on the street in the exhibit hall. We loved chatting with people that are living with hemophilia B, that are treating hemophilia B, and that are researching future science, in particular for hemophilia B. So thank you to CSL Behring.
It's a magical world. And stay tuned with us up next, where we will hear from Colorado's, once again, a shout out Colorado, Kaylee Dollar-Shell, their genetic counselor, and the importance of genetic counseling with your gene therapy. So
On the next segment,
stay tuned. It's your girl, Hannah Berry.
And Amy Board.
Bye, friends.
Bye.
Thank you to everyone who stopped to speak with the wonderful Amy Board and Hannah Berry during BDC 2026. That was quite a whirlwind of voices that we got to hear from. I will. That was a whirlwind. I absolutely will. And again, thank you to CSL Behring for supporting today's episode and our gene therapy segment on the BloodStream podcast. We literally could not produce this show without your support, and we are grateful for it. Bordeaux, next episode comes out October 24th. I cannot believe October 24th is a date that's coming out of my mouth. What can listeners expect to hear?
We have an interview with Patty Welton.
Who dat?
Who dat? She's actually curating an art installation with children that have rare diseases, that are overcoming challenges with rare disease, and our very own BloodStream family member, Effie Parks, her son Ford, is featured in this art installation. And so you have a lovely conversation with Patty. Oh, that's kind of cool.
Yeah, I saw the installation at a New England Hemophilia Association family education weekend, like pre-COVID, a long time ago. And it struck me then, and it's always been on my radar. And this was the first time that I reached out to, I even said to her, like, I think this is long overdue. I'm sorry I haven't done this sooner, but I'm Patrick and let's talk for BloodStream. So she said yes, we had a lovely conversation. That's coming up on the next episode. And with that, that is all for this episode. Be sure to subscribe to BloodStream Podcast wherever you get those podcasts to have the next episode delivered to you the moment it goes live.
And loyal listeners, as always, email us at mailbag@bloodstreammedia.com to inquire about our casting opportunities or to share your story. We love that. We love hearing from you all, and we love your ideas about segments and guests and certain things. So if there's a gap or something that we're missing, please let us know. And you can also find us on social media. You can find Believe Limited, you can find BloodStream Media, and me and Patrick.
It's amazing the options that you have.
Literally the options.
I'm your host, Patrick James Lynch.
And I am your other host, Amy Board.
And until next time, take self-care of yourself. Bye, everybody.
Bye-bye.